Our team made up Olga Daniela, Fernanda Jimena Hernández Pinto, Antonio de Jesus González, and Miguel Cabrera are going to explain the methods and techniques used for the sequencing of nucleic acids, both chemical and enzymatic methods. Describe their main applications and research impact that these two methods have had in history, and get to know the laboratory equipment used in these technologies. In modern molecular biology, DNA sequencing, known as the ability to determine the sequence in DNA, is such an important part in this discipline. In the late 1970s, two rapid sequencing techniques were developed, the chemical and enzymatic methods, and now those revolutionized and improved techniques are used in laboratories worldwide. Now, I'm going to explain the chemical method for sequencing DNA developed by Alan Maxam and Walter Gilbert. In this Maxam and Gilbert method, chemicals are used to clip the DNA at certain positions, generating a set of fragments that differ by one nucleotide, and a defined fragment of DNA is required as the starting material. In order for the technique to be applicable to any DNA fragment, the starting material need not to be cloned in a plasmid vector. Then, the DNA is radiolabeled with 32 phosphorus at the 5 ends of each strand, and the strands are denatured, separated, and purified to give a population of labeled strands for the sequencing reactions as shown in the next slide. Now, the next step is a chemical modification of the bases in the DNA strand, which is done in a series of 4 or 5 reactions with different specificities, and the reaction conditions are chosen so that, on average, only one modification will be introduced into each copy of the DNA molecule. Finally, the modified bases are then removed from the sugar groups, and the strands cleave at these positions using the chemical piperidine.
Sanger-Coulson sequencing, also known as dideoxy or enzymatic sequencing, was developed in 1977 by Nobel laureates Frederick Sanger, A.R. Coulson, and colleagues. This was a breakthrough at the time due to the special use of dideoxy nucleotides. First, the DNA to be sequenced has to be amplified. The DNA sequence of interest is used as a template for a special type of PCR called chain termination PCR. Chain termination PCR works just like standard PCR, but with one major difference: the addition of modified nucleotides, called dideoxyribonucleotides. In the extension step of standard PCR, DNA polymerase adds DNTPs to a growing DNA strand by catalyzing the formation of a phosphodiester bond between the 3,3-OH group of the last nucleotide and the 5-phosphate of the next one. The second step, the chain-terminated oligonucleotides are separated by size via gel electrophoresis. In gel electrophoresis, DNA samples are loaded into one end of a gel matrix, and an electric current is applied. DNA is negatively charged, so the oligonucleotides will be pulled towards the positive electrode on the opposite side of the gel. The last step involves reading the gel to determine the sequence of the input DNA, because DNA polymerase only synthesizes DNA in the 5' to 3' direction, starting at a provided primer. Each terminal DNTP will correspond to a specific nucleotide in the original sequence. Therefore, by reading the gel bands from smallest to largest, we can determine the 5' through 3' sequence of the original DNA strand.
Modern DNA sequencing began in 1977 with the development of the chemical method of Maxam and Gilbert, the enzymatic method of Sanger, and with the first complete DNA sequence. Since then, many new techniques and equipment have been developed in order to quicken, automate, and cheapen the process of DNA sequencing. One example of such includes the Pacific Biosciences sequencing technology that was launched in 2011 under the name SMART, which stands for Single Molecule Real Time. Basically, how this technology works is by taking isolated DNA or RNA from any sample type and creating a library by ligating adapters to double-stranded DNA, thus creating a circular template. Primer and polymerase are added to the library, and the library is placed on the instrument for sequencing. The sequencer holds a SMART cell, which contains millions of tiny wells that are called zero-move waveguides, or ZMWs. Single molecules of DNA are immobilized at the ZMWs. Then, stranded DNA is threaded to the polymerase, while fluorescent-labeled nucleotides are incorporated and individual color is detected when cleaved with the pyrophosphate group. Light is emitted and a CCD camera takes video as the DNA gets polymerized. Incorporated nucleotides are detected based on the associated fluorophore that is released and dissipated upon cleavage of the phosphate chain. Smart sequencing covers a broad range of applications, including whole genome sequencing, variant detection, RNA sequencing, to resolve complex populations, targeted sequencing, and epigenetics.
One of the striking aspects of the gene manipulation technology is that many of the procedures can be carried out with a very basic laboratory setup. General facilities include aspects such as laboratory layout and furnishing provisions of essential services such as water, including distilled and/or deionized water sources, electrical power, gas, compressed air, vacuum lines, drainage, and so forth. Most labs will require facilities for growing bacteria cells with the need for the equipment such as autoclaves, incubators, statics and androtory, centrifuges, and protective cabinets in which manipulation can be performed. Mammalian cell cultures require slightly more sophisticated facilities, and plant or algae culture usually requires integration of lighting into the cultural cabinets. Every gene manipulation experiment requires a source of nucleic acid in the form of either DNA or RNA. It is therefore important that reliable methods are available for isolating these components from cells. There are three basic requirements. One, opening the cells in the sample to expose the nucleic acid for further processing. Separation of the nucleic acids from the other cell components, and the recovery of the nucleic acids in purified form. A variety of techniques may be used, ranging from simple procedures with few steps up to more.
Our team concludes that at the rate that science and technology are evolving, there's no doubt that new and better sequencing technologies will emerge. However, both the Sanger and the Maxam and Gilbert methods were very important milestones that helped build the foundations for DNA sequencing.